Hello, class. In this lecture, we will be introducing phylogenetics and discussing how to read a phylogenetic tree. So the very first question is, what is phylogenetics? And phylogenetics is the study of the evolutionary relationships between biological entities. So usually when we think about phylogenetics, we're thinking about like how are different species related, right? So how are species A and B related, and where was their common ancestor, and so on and so forth. How have they evolved? But we can also apply this to genes as well and the proteins that they code for. And we are studying phylogenetics by using phylogenies or phylogenetic trees. And these are going to be visual representations of these evolutionary relationships. So throughout the course, you may hear us say the word phylogeny, phylogenetic tree, or just tree. All of those are synonymous. So if you hear any of those, they all mean the same thing. And on the left here, we have a schematic of a very just general phylogenetic tree. So let's break that down and discuss the parts of a phylogenetic tree. So over here on the right-hand side of the phylogeny are going to be our species of interest, or they're known as tips, so the tips of the tree. And these are going to be our taxa of interest here. And so we mentioned this earlier in the course, is that taxa are going to be like any group of organisms, so whatever you're studying. So that could be species, that could be genera, that could be families, whatever the realm that we are looking at, that is what we're talking about with these species of interest. So in connecting our species of interest are going to be branches. So these, right in this phylogeny, they are horizontal, but these horizontal lines are going to be our branches. And what these are showing is going to be changes over time. And so the closer we are to our tips, the closer we are to present day. And then vice versa, the further we are from our tips, the more backwards in time we are moving. Connecting our branches are going to be what are known as branch points or nodes. That's what we use more commonly, is going to be a node. And this represents a common ancestor in that period of time, or in that period of change. So for example, right where my pointer is hovering at, this node right here, this represents the common ancestor of species C and D. So this is where we have a common ancestor, and then that ancestor diverged and split and became species C and D. If we go a little bit further back, this node right here, where my pointer is at, that is going to be the common ancestor for species A, B, C, and D, because it is connecting all of them right there. So that is the last time that all four of those species, or all four of those taxa, had a common ancestor. If we go a little bit further back, then we get to what is the root of our phylogeny. So this is going to be the last common ancestor of all taxa that are in our phylogeny. And so what we are trying to make, quote unquote, make when we're looking at our phylogeny is going to be clades. So those are going to be groups of organisms that have evolved due to a common ancestor. So I already mentioned this node right here where my pointer is at, that if we include this ancestor and then all of its descendants, that would be a clade right there, because all of these A, B, C, and D have all evolved from the same common ancestor, so they would form a clade. So usually we want to form what is known as a monophyletic clade. So a monophyletic clade is a common ancestor and all of its descendants. So if we take a look at this phylogeny that is shown right now, this is going to be a phylogeny of all vertebrates. And so we can see what characteristics are being used in order to determine what new groups we are looking at in this phylogeny. And if we take a look at the yellow group right in here, this yellow group is including all of our reptiles and then also birds. So what this is showing is that reptiles and birds are a monophyletic clade. They all share one common ancestor. If we were to say, let's just take a look at reptiles and reptiles only, then we are looking at this like teal blue color right here. And so that would be that would be including our common ancestor and some of its descendants because we are looking at reptiles only and not birds. And so that would be a paraphyletic group. So contains some, but not all of the descendants of a common ancestor. And then if we wanted to discuss, say, birds and mammals, like what this red color is showing. So here's our mammals and our birds. They share a common ancestor way back here at the beginning of Amniota. Then this would be a polyphyletic group because they are two groups that do not share a most or more recent common ancestor. So when we're interpreting our trees and when we're making our like taxonomic groups or making some assumptions, we're really looking for monophyletic clades. We usually want to look at what like the most recent common ancestor and then all of its descendants. But when you guys are interpreting your trees, you may end up with paraphyletic or even polyphyletic clades when you are looking at the taxa in your protein trees. So I have a bit of a question for you. So going back to our previous schematic of a phylogeny. So looking at this phylogeny right here, which of the species of interest, so which of the tips would be the most primitive? So would that be A, B, C, D, or E? So you may have said that E would be the most primitive species of interest, and that would actually be incorrect. There is no primitive species of interest in this phylogeny, and that is because we are looking at this like through time, right? And so species E is evolving just as long as species A is. It's just A has more species that are more similar to it, and E is less similar than all of the other species. So there is no more primitive in phylogenetics. There is just more or less similar. So organisms that are going to be at the bottom of a phylogeny or like organisms that seem like they are more separate from all of the other organisms, they are not more primitive. They do not have primitive characteristics or anything like that. They just are less similar than all of the other included taxa. So if we added more taxa in, so in this case, species F and G, now that might like trick your brain into thinking that F and G are now more primitive than E is. But that's just showing you that as we add more taxa, we're just adding to the information that we know. And so now E is no longer like at the bottom of the phylogeny. It is more similar to species A, B, C, and D than it is to F and G. All right, so there's no more primitive or ancestral extant species. All right, they've been evolving for the same amount of time. So why would we want to study phylogenetics? So we would like to study, or we want to study phylogenetics because helps us understand many different biological aspects. So how have organisms evolved? Like how have species evolved? How have proteins and the genes that code those proteins, how have those evolved? Where have those proteins come from? So on and so forth. How have physical characteristics evolved? How have physiological characteristics evolved? Things like that. So we have these questions, and phylogenetics helps us answer those questions. And this is really important for a variety of other biological research, including biodiversity and conservation. So in this course, we are going to be focusing on biodiversity, right? So what organisms are around in the world, and in where are they? What are they doing? So on. But it's important for conservation because if we understand who as in what organisms are in different areas, do they need protecting? How are they all related? Things like that. One that you may have come across in the more recent years is actually disease and pathogen evolution. So this phylogeny shown here on the right-hand side is a phylogenetic tree of COVID, right? Of the most or of the COVID epidemic. And so what this is showing is going to be the different strains of that COVID viruses that were sampled at that point in time. And what they were doing was they were tracking where the different strains were coming from and how they were evolving in order to get our different vaccines and things like that. So phylogenetics is relevant to health research as well. We are going to be doing protein evolution, right? So how have proteins evolved? Where have those proteins come from? And how might they be helping the cells that they are in, right? So phylogenetics is going to give us an insight to our protein function as well. So just kind of going back to more basics about phylogenetics in how they are constructed is phylogenetic trees are going to be built by comparing characters shared by organisms due to common ancestry. So we're going to assume that is due to homology, all right, due to common ancestry. And so we can compare multiple different characters. So for example, we can compare anatomical features. So this phylogeny on the right-hand side is showing a compare or using anatomical features in order to make the phylogeny. So we are going to group our organisms into clades based on what anatomical character they have. So for example, if they have four walking legs, then we're going to bring them all into one clade. If they have hinged jaws, things like that. And so that what you end up with is that at one point in time, one of these characters had been acquired, and then uh, all of the progeny afterwards have that character. So, for example, we have the gain of the vertebral column here, which means that right here, right before this node, which means that after this, all of the organisms have a vertebral column, and then anybody that's not included in that with that character is going to be outside of that group. So landslets, which do not have vertebral columns, they are going to be outside of this group, and then everything else is going to be vertebrates. And we can just keep going with that. So if they have hinged jaws, then they are going to be included in a group, in a clade. And then if you don't have a hinged jaw, but you still have a vertebral column, then you are going to be in your own little your own lineage, such as our lampreys right here. And we can just keep doing this in order to build up our phylogeny. We can also use physiological features like whether or not they have certain functions or whether or not they have certain proteins or enzymes in order to create our phylogenetic tree. But the more common, especially right now, is going to be using molecular features. So those can be either nucleotides or amino acids. So if we are just going to be building like a quick barcode phylogeny of our organisms, we might use a gene such as the small subunit, the small ribosomal subunit, which is also known as the 18S in eukaryotes or the 16S in bacteria. And so we can use those nucleotides, that gene of interest, in order to make a phylogenetic tree using that gene. Or we can also use amino acids in those cases, or which would lead to the proteins that those amino acids are going to create. And so that is what we are going to be doing in our class, is we are going to be using amino acids that we have pulled from our organisms in order to construct phylogenetic trees. So we've been mainly showing some really basic trees, right? But there are a couple examples of some non-basic looking trees. But the ones that we've shown you where we have branches that are all leading up to like they're all the same length, leading up to like a nice straight line of where all of our species of interest are. Those are going to be cladograms, and what they are showing is going to be taxonomic relationships only. And this is just for ease of being able to read, right? And so what this cladogram is showing us is that A and B are more similarly related than they are to C. And that's also shown right here in this other cladogram. So A and B are more similarly related compared to C. But again, this is just showing taxonomical relationships. If we want to also look at the rate of evolutionary change, then we will end up with a phylogenetic tree. And that is that is when we get our different branch lengths. So our tips are still going to be extant species, so still species that are around today, even though they're not landing at the same like level. But the length of the branches is going to equate to the amount of evolutionary change over time. So the longer branch you have, the more change you have. The shorter branch you have, the less change you have. So now we are not only looking at who is related to who, but also how much change has happened that is separating all of these different organisms. So in this case right here, this long branch on the left-hand side, not only is it is it dissimilar than all of the other species of interest, but also it has this really long branch to it. So it has had more evolutionary change over time compared to the others. So one of the last things that we're going to bring up is going to be like kind of how to interpret your trees. And so that's what tree thinking is. Tree thinking is, how do we reason through our phylogenies? How do we interpret our phylogenies to understand what it is telling us? And so this will lead us to be able to answer our evolutionary hypotheses of what's going on evolutionarily, right? So how have things changed, or where did these different proteins, these different proteins of interest come from, so on. So we're going to be looking at patterns. And the basis of this is just to follow the most recent common ancestor between our taxa of interest, okay? So I'm going to introduce this phylogeny down here. So we have, it's actually a cladogram, right? Because all of our branch lengths are leading to the same like horizontal level, right? So introducing this phylogeny up here, we have pine and moss and green alga, red alga, and then amoeba. So the question is, which of the following is an accurate statement of the relationships within this phylogeny? So is green alga more closely related to red alga than it is to a moss? Is green alga more closely related to a moss than it is to a red alga? Is green alga equally related to a red alga and a moss? Or is green alga related to a red alga but is not related to moss? So let's break down each one of these statements. So green alga right here is more closely related to red alga than it is to moss. So let's follow our branches to our nodes, to our last common ancestor, right? So if we follow the green alga down, here is that node. This is our last recent common ancestor for green alga, and it is sharing that last common ancestor with this clade, including moss and pine, right here at X. Now, if we look at the last common ancestor for green alga and red alga, it is going to be down here at Y. So following that branch, those branches down to Y. So this first statement, green alga is more closely related to red alga than it is to moss, would be incorrect because green alga and moss share a more recent common ancestor than green alga and red alga. So the second statement, a green alga is more closely related to moss than a red alga. We kind of just answered that by breezing our way through A. So green alga is more closely related to moss than it is to red alga. That would be true because green alga and moss share a more recent common ancestor than red alga and green alga. C, a green alga is equally related to a red alga and a moss. So green alga equally related to red alga and a moss. That is incorrect because they share different last common ancestors. And then lastly, a green alga is related to red alga, but is not related to a moss. So green alga is going to be, again, more closely related to moss and pines than it is to red alga. So this is a general example of tree thinking, reasoning our way through the tree in order to figure out who is more closely related between our taxa. So we can use tree thinking and then everything else that we've discussed to kind of answer different questions. So things like how do different behaviors arise? How do morphological changes occur? How do organisms occur in different areas of the world or in the same area of the world, which is biogeography. And so we are going to use our tree thinking and use our background knowledge of our organisms in order to answer those questions, okay? So we are going to be making inferences about our organisms using the phylogenies that we make and our background on their biology. So we can use phylogenies to make inferences about, like, for example, we can use phylogenies to make inferences about termites and their protists. All right, so in this case, what this is showing right here is that a termite in the black is going to be a termite lineage, and it is going to be diverging. And then in the blue is going to be a protist lineage, and then it is also diverging. So an inference that we can make is that as the termites diverge and evolve, so are the protists that are living in it. All right, so that's like what this is showing us right here. We can make other assumptions about termites and their protists by using different phylogenies. So, for example, this one right here. So, on the left-hand side is a cladogram of termites in termite families. And then its outgroup is going to be Cryptocercus, which is a wood-feeding roach. And Cryptocercus, and then almost all of our termite families have wood-digesting protists within their hindguts. And then there is one family termitidae that is protist independent. It does not use protists in order to digest wood and gain nutrients. So with that, the question that I'm asking is, what inference can you make about protists and termites with that background information? Well, considering that Cryptocercus and most of our termites have wood digesting protists within them, we can assume that the ancestor, like prior to the ancestor of termites and Cryptocercus, they had to have had protists, right? Because that would be one step of gaining protists rather than having multiple steps between Cryptocercus and termites having these wood digesting protists. So they most likely entered into the guts of a ancestor of Cryptocercus and termites. And then, since all of our termites except for one clade have protists, the other assumption that we can make is that this clade right here, Termitidae, has lost their protists. That would make more sense than having a whole bunch of introductions of protists into termites. So using the biology of which termites have protists and which outgroups, so for example, Cryptocercus have protists, and we can make inferences to when those protists have entered into the system, entered into a symbiotic relationship with these insects, and when they were lost. So using phylogenetics and our background knowledge. So here's another example right here. On the left-hand side, we have a phylogeny of Reticulotermes, which is a genus of termites, and it is color-coded based on the map that is here on the right-hand side, and so showing where those termites are from. So up here in the top, we have Reticulotermes that are from Europe and in Northern Asia, and then also from Southern Asia and Central Asia. And then here in this bottom clade, we have Reticulotermes mainly from North America, with one instance from Europe. So if we're looking at this phylogeny, what inferences can you make about Reticulotermes and where they occur, right? Let's just take a moment for that. So if we're looking at this bottom clade here, all of these Reticulotermes except for one, which is nested within all of the other Reticulotermes, is from North America. So we can assume that all of these Reticulotermes originated in North America. And then since there is this one that is found in Europe, the assumption can be made that there was some sort of event which brought that North American Reticulotermes to Europe. And then up here on the other side, up here in this other clade, all of these are from basically Eurasia, right? And so we can make the assumption that the ancestor to this clade originated in Eurasia. And so with all of that background, we can assume that in the ancestor to all Reticulotermes, there was a split. And so that some a lineage of Reticulotermes is going to be in North America, and then a lineage of reticulotermes is going to be in Europe and Asia. So again, we are using our background knowledge on where these termites are from in order to make inferences as to the evolution of these taxa. So the last thing that we're going to discuss is going to be very relevant for you guys, and that is going to be a species tree versus a gene tree. So gene trees are going to be trees that are constructed based on one individual gene. All right. So down here in this little schematic of different cladograms are going to be 4 different gene trees, all from 3 of the same species. So species A, B, and C for all of them. But we end up with different tree topologies, so different ways that the species are related to each other based on which genes we are using. So up here in the top part, locus 1 and 2, that is or loci 1 and 2 for plural. These are showing us that species A and B are more similarly related at this locus than they are to C, in comparison to loci 3 and 4, where species B and C are more similarly related than they are to A. And so we can't take one of these trees and go, oh, I know that A is more similarly related to B than it is to C, because we end up with different tree topologies based on which gene tree we are looking at, right? So in order to make a species tree, what we're doing is we are bringing in all of the information that we have in order to make a consensus tree, in order to make a tree that that is taking in as much information as possible to give us what is the true species tree. So in this case, the species tree is going to be A and B are more closely related versus C. All right. And then within our species tree, which is outlined in black, we can see these individual gene trees and how they may be similar or maybe different compared to our species tree. So in this class, we are going to be working with species trees. I'm so sorry. We are going to be working with gene trees. You are going to be building individual gene trees, and then we are going to be comparing them to a schematic of a species tree. So we're not 100% sure what the species tree is for our metamonads. That is a ongoing research project, but we can make our best guesses to it. And then hopefully here in the near future, we will have a good species tree for metamonads that we can compare our gene trees to. So the last thing that we are going to be discussing is going to be our gene trees, like what they may look like. So we are going to be comparing homologs, all right? So homologs or homologous genes are going to be genes in different organisms which arose from a common ancestor, all right? And so any kind, yeah, like any kind of common ancestor lineage sorting deal here. And we can further break up homologs into orthologs and paralogs. So orthologs or orthologous genes are going to be homologs that are in different organisms due to speciation. So if we follow our genes here in this example, so gene A, it is going to go through some sort of speciation event, and each one of those genes, each gene is going to end up in different organisms. So in this case, we still have gene A, but it is in a human and a mouse. So those are orthologs because it's still the same gene. It's just now we have them in different organisms due to speciation. And that happened over here in gene B as well. If we have a paralog, that is when we have the duplication of a gene, of an ancestral gene somewhere. And then we end up with basically two different genes that originated from the same one. So one of them may have lost their original functionality or lost function altogether, but now they are two separate genes that have ancestry from a single gene. So we can see our ancestral gene up here in blue, and then there was a duplication event, which led us to gene A and gene B, which can still be traced back to this ancestral gene. And then each one of those genes went through speciation. And so now we have gene A in our organisms and then gene B in our organisms, both in humans and in mice, right? So now gene A and gene B are going to be paralogs because they are in the same organism, but we have multiple duplications of that same gene. So you may come across these paralogs when you are looking at your phylogenies, and so that may look like duplicates of that same gene or of that same protein. So just be aware of that. But we're really hoping for is to end up with a ortholog phylogeny for each of your proteins. So that is the introduction to phylogenetics, and we will discuss your phylogenies more in depth in later on in the class.